Alright, many characteristics of pea plants are genetically controlled. One of these characteristics is flower position. Flower position can be either axial or terminal. In a first cross, scientists crossed pea plants with axial flowers with pea plants with terminal flowers, and it led to a cross that produced only axial flowers, produced 1,120 offspring plants, but they were all axial flowers. And it wants us to use the genetic diagram to show the genotypes of the parent plants, the gametes they produce, and the genotypes of the offspring plants. Now, before we do anything here, we need to decide what our letters are going to be for our gametes and for our Punnett square. And just for simplicity, I'll just use A. Big A, little a, right? This one is dominant, recessive. Okay, now what could the genotypes be for the parents? Because we only know their characteristics, right? Well, if you actually read this, you should see that since we're crossing them, right, and they're producing only axial flowers in the offspring, we should know that axial is going to be the dominant trait. It's going to be the capital letter. Now, the way this works is if we cross a homozygous dominant with a homozygous recessive, we'll see what happens with the Punnett square, but this is essentially the combination we have. This recessive is going to lead to the terminal flower position, while the homozygous dominant is going to lead to the axial flower position, and that's going to produce only axial flowers. Now, let's do a Punnett square to show the gametes they produce. So we have something that looks like this. Okay. So we have this homozygous dominant, and we have a homozygous recessive. Now let's see the gametes they produce. Heterozygote, another heterozygote, another heterozygote, another heterozygote. That's going to be 100% heterozygote. What this means is 100% of the dominant phenotype. I mean, the dominant phenotype is axial flowers, which perfectly explains the data we have. So it's going to be 100% hetero, okay? And this is essentially these are the offspring, right? They're all going to be heterozygous, right? And we know the gametes formed, well, the gametes formed we can see over here in our Punnett square, all visible. And yeah, we can get all marks just from showing them in a Punnett square. So pretty simple question, easy to get. There we go. Okay, the scientists allowed this offspring from the first cross to self-fertilize. Second cross produced 858 second-generation plants. 608 had axial and the others had terminal. Calculate the ratio of plants with axial flowers to them with terminal flowers, n to 1. Okay, so first things first, let's calculate how many had terminal flowers by doing 858 minus 608. Okay, that gives us an answer of 250. Okay, so then we know there's 250. We know the ratio is 608 to 250, but we want it in the form n to 1, so we just divide it by 250 to get n to 1, and then it goes 2.43 or 2.4 to 1. Okay, there we go. That's essentially what we're going to do. Okay, explain why the ratio of plants with axial flowers to plants with terminal flowers is different from the expected ratio, right? So this isn't the expected ratio that we expect when we're doing the offspring from the first generation to self-fertilize. The offspring all are heterozygotes, remember. So if we were to do it, I'll just show you. It would be something like this, right? And then we would get something like this. Okay, so that means that although they're different genotypes, that means 3/4 of them are going to show axial, and that 1/4 are going to show the terminal. But in reality, we have a different to the expected ratio. We have something different. It's supposed to be 3 to 1, right? 3 to 1 ratio. But instead, we have 2.43 to 1. So why is that happening? Why is it different? Well, first, let's write 3 to 1 is expected. And the reason for this is because there is a role of chance. There's a probability. Okay, nothing is guaranteed. These are just the expected odds, right? And this happens during fertilization, right? There's always chance in fertilization. Now, if you know about crossing over, random orientation, you should know why this occurs. But if not, just know that there's a role of probability in fertilization. And this is essentially why we're getting a different ratio to the expected. And another reason if you didn't want to go this path, is say that there's fewer axial flower positions that survive, or that are pollinated, and that's probably why there's less. But that's a bit of a stretch, but it's in the mark scheme, so it works. Okay. All right. Next up, we have a quorums question. So this is essentially a design investigation question. If you don't know what quorums question means, it's basically a technique you can use to answer this question. There's a question like this in every paper one, essentially, and the question always has things that are common with each other. Like, it's 6 marks. It asks you to design an investigation. It gives you context, but you don't really need any prior knowledge to answer the question. And it's very simple. You just need a technique to answer it. And I won't be answering this in this video because I have a very detailed, good video on how you can answer any quorums question, and that would help you a lot more. So I'll leave the link to that video in the description. But yeah, after you watch that video, if you want to try out this question yourself, you can try it out, and hopefully you'll be able to answer it. So next one. Okay, lipase digests lipid into fatty acids and glycerol. The fatty acids change the pH of the solution. A student uses this method to investigate the effect of temperature on the activity of lipase. Sure. Here's the method. I'll look at it later. Yeah, it's quite long, so I don't want to read it right now. The student repeats this method for 5 more temperatures and those temperatures. Sure. Okay, it's asking us, state why the student leaves the test tube and beaker in the water bath for 5 minutes in step 6 and 7. So 6 and 7, it's asking us to put the test tube. The test tube is filled with what? Let's see. Let's read steps 1 to 6 here. So we have a test tube labeled 20 degrees Celsius. Add 5 drops of phenolphthalein indicator to the test tube. Add 5 cm cubed of milk. Add 7 cm cubed of sodium carbonate solution. Place a thermometer and place the test tube. Sure. So we can see here the test tube is for the 20 degrees Celsius condition. So why are we putting it there? Well, we want the test tube. We want so the test tube contains a substrate here, right? It's contained in the substrate. But we want the test tube to be the correct temperature, but it's also asking us why we're wanting the beaker to be in the water bath as well at 20 degrees Celsius. Well, we're adding the enzyme to the solution, right? So we want that both the enzyme solution and the milk solution that we made, we want both of them to be the appropriate temperature. So we want both test tube plus beaker solutions to be at the correct temp, right? We don't want to say optimum temp because it's not really optimum. We're just picking the correct temp for our specific part of our investigation. We're doing 20 degrees Celsius now, so that's the one we're investigating, so we're getting it to that temperature. If we're doing 40 degrees, then we're looking at 40 degrees. Water bath's going to be at 40. So you can see that we just want the correct temperature. We're not looking at optimum here. Okay. Give the dependent variable in this investigation. Let's have a look. It should be in step 10. Stir the contents and record the time until the contents lose their pink color. And yeah, that's essentially what we're measuring. So time till contents lose pink color. There we go. State one variable the student controls in the investigation. Let's see. Well, there's many things here. They might not be completely explicitly said, but we can see them by just inferring them from the steps. So first thing is that the volume of lipase, it's fixed. He's using a fixed volume of lipase. What else? Well, the volume of milk, we can see 5 centimeters cubed of milk. That's a control variable. So I'm just going to use one of those. 5 cm cubed of milk used across trials. That's a control. We see the same volume of milk. You can see that for the rest of them as well. Same volume of sodium carbonate, whatever you want to use. Okay. So just the purpose of the phenolphthalein indicator. Well, we didn't really need to look at the rest of it, the rest of the method for that. We can just answer it. And the reason we're using phenolphthalein is to show that all the lipid is been digested, that has been broken down. So it's our indicator. Show that all lipid has been broken down. Simply done. Okay, the table shows the results. We have temperatures and the time taken. Sure. Plot a line graph to show the effect of temperature on the time taken. Use a ruler to join your points with straight hands. Now, I'm going to leave this question here like that. It's quite difficult for me to draw a line graph right now because, yeah. Explain why increasing temperature affects the time taken for the contents of the test tube to lose the pink color. Okay. So, okay, explain why increasing temperature affects the time taken for the contents of the test tube to lose their pink color. Classic enzyme question. Should be free form amongst you guys. Very easy. So first thing first, increased temperature is increased. I'm going to write in symbols and abbreviations, okay, just for the sake of time. So increased temperature is increased kinetic energy, right? Ke. And this means that the molecules move faster, okay? So what does this actually do? Well, this means that more enzyme substrate complexes formed, okay? More successful collisions, more frequent successful collisions. Now, what does this actually result in? Well, this means that the time taken for the contents to lose color, time taken, okay, is lower. It decreases, right? Because there's more reaction. The reaction's occurring faster. Now, we're going to have to answer this with nuance, though. We can't simply write that because it's saying, explain why it affects the time taken for the contents to lose the pink color. We have to explain what happens to the optimum, because if we keep increasing temperature, it's not going to exponentially increase. There's a point where it starts to decrease. So we have to mention that. So we have to say that this occurs, all of this occurs, this increase in reaction rate, this occurs until optimum reached. Now, once we've reached the optimum, right? Once we've reached that level of optimum, what's going to happen after? After the optimum, after denaturation occurs. Okay, denaturation occurs. This means the bonds in the active site break. There's a change in the active site. So what this means is the substrate can no longer bind to the enzyme. And there we go. That's more than enough marks. Yeah, pretty simple. Yeast can be used in experiments to investigate the effect of different concentrations of glucose solution on the rate of anaerobic respiration. Yep, can. And the diagram shows the apparatus used to measure the rate of anaerobic respiration in yeast. So we have a nice little apparatus here. Now, okay, and it's asking us to give the function of the water bath. Interesting. So without even knowledge of this apparatus, which you probably won't have good knowledge of this apparatus, but without knowledge, you should be able to infer what the point of the water bath is. So why would we need a water bath? Well, we want to maintain temperature. We want to keep controlled environments, so maintaining temp. There we go. Okay, liquid A can be used to identify the gas released during anaerobic respiration by the yeast. Explain which substance can be used as liquid A. So first, before we look at liquid A, let's think about what gas is released. Well, we should know that it is carbon dioxide. During anaerobic respiration, yeast is going to release a gas, which is carbon dioxide, right? The gas it releases is carbon dioxide. Now, what can we use to identify carbon dioxide? Well, you should know from your practicals that it's lime water, CO2. And we can use lime water used, and the way we use it is it turns cloudy in the presence of CO2. There we go. What else can we say? Well, if you are very clever, you should know that we can also use hydrogen carbonate or sodium hydrogen carbonate. And what happens, it will turn yellow or slightly orange in the presence of carbon dioxide. But this is the easy one, CO2 lime water. It's very simple. Okay, a student wants to accurately measure the rate of anaerobic respiration in yeast using this apparatus. State what additional apparatus they would require. So we're able to measure, we're able to identify this change, but this is just to see if carbon dioxide is present. We can't actually measure the amount. So we'll need to get the volume of carbon dioxide. We're going to need a gas syringe. can also use a measuring cylinder, use displacement, but gas syringe is easiest. What else can we use? Well, we need to measure rate. Remember rate, and something you always need for rate is time. So we're also going to need a stopwatch. Very easy apparatus to name. There we go. Okay, the process of anaerobic respiration is sometimes referred to as fermentation. Industrial fermenters are used to grow microorganisms. These microorganisms produce penicillin, an antibiotic that is used to treat bacterial infections. Explain how named conditions in an industrial fermenter are controlled. Pure theory question. I think this is just based on your understanding. If you've studied hard enough, you should know everything. Okay, so how are the named conditions in the industrial fermenter controlled? Okay, first named condition, an aerator, right? We can have an air inlet, I believe Sparger is also one of them. And what is the purpose of an aerator, right? It's to provide oxygen for all respiration, for respiration to occur. For respiration. You just need 4 here. What else can we say? Well, we would need a pH probe, pH monitor, something to measure the pH, right? And why do we need that? Well, we need to maintain optimum pH, right? We need to maintain this optimum pH for the reaction to occur. And just like that, we can also think we need some sort of temperature monitor. Temperature monitor, okay? And well, how do we use that? Well, we would use it to maintain optimum temperature, right? We want to obtain the optimum temperature that we need, prevent it from overheating or becoming too cold. We need an optimum temperature for the reaction to occur. Yeah. Okay, and fourth and final one, what else can we use? Well, an important thing you might have remembered is an air filter. It's quite noticeable in fermenters. Oh, sorry, I wrote noticeable, sorry. We need an air filter. An air filter. Now, what do we use it for? Well, the reason we use it is to prevent contamination, right? We don't want bacteria to be entering because if bacteria enters the fermenter, it will grow because the conditions of the fermenter are meant to be suitable for things like bacteria to grow microorganisms. So we want to prevent any contamination of any external things, external pathogens, external bacteria. And some other things you might want to mention is that you can have stirrers inside of a fermenter. And the reason for them is pretty self-explanatory. We use stirrers to stir, to mix the contents of the fermenter. Okay, some antibiotics are becoming less effective at controlling bacterial infections. Explain how bacteria have evolved so antibiotics are less effective. Pure antibiotic resistance question should be very simple for you to understand. So the basic logic behind this is that a mutation occurred in the bacteria. Right? Bacteria multiply a lot. So one day a certain mutation occurs. Okay? So mutation occurs in the bacteria. And this mutation somehow luckily means that this bacteria has a resistance to antibiotics. To an antibiotic. Okay? So because it has this resistance, when we use antibiotics, it means that these are the only bacteria that survive. The rest of them are killed by the antibiotics, but these resistant ones survive. So resistant ones survive. Now that they survive, since they're able to survive, they're able to reproduce. They're able to produce many copies of that bacteria via binary fission, and that means that many bacteria have this resistant gene. So resistant bacteria multiply. Okay? Now that they multiply, they're essentially passing on the gene, their alleles. They're passing it on to future generations. And this means that there's an increased frequency. This is an arrow, by the way, it just means increased frequency. Oh wow, frequency of that gene, the resistant gene. And this is going to cause major problems and mean that more bacteria are antibiotic resistant and antibiotics overall are less effective. Okay, GM bacteria are used to produce insulin. Explain how GM bacteria are produced. Oh, these GM bacteria, so these insulin-producing bacteria. Okay, this is just very easy question. It's, again, just listing out the steps. You should be able to remember it. So first thing first is we use a restriction enzyme. Specifically, it's a restriction endonuclease enzyme, but we can just say restriction enzyme. So we're using a restriction enzyme to cut the gene, the insulin-producing gene, right? We're using this restriction enzyme to cut it. And then after we cut it out, we're going to have to use that same restriction enzyme. So use the same restriction enzyme to cut a plasmid. The plasmid is this organelle of bacteria that's essentially like a circular loop of genetic material, and we can use it later on. We're going to see how we use it, but essentially we're using it as a vector, essentially a vehicle to transfer our gene to the bacteria. So cutting it with the same restriction enzyme, it basically allows us to attach the cut gene to the plasmid. If we use a different restriction enzyme, think of a Lego piece that doesn't fit with each other. It won't stick to each other. It just won't work. So that's why we use the same one. Okay. Now, what do we do? So we can use lipase enzyme to attach gene to plasmid. The plasmid, and now we have to attach our puzzle piece that we cut out from the gene. We're going to cut out this gene that produces insulin, and we're attaching it to the plasmid. And ligase here is acting like a glue. It's essentially glue. Okay. There we go. And now, after we've done that, it just comes down to simply reinserting this plasmid, our vector, back into the bacteria. So insert plasmid to bacteria. And now that plasmid's inside the bacteria, and it has that new genetic material, and it's able to produce large amounts of insulin, which is wonderful. It's a great way of producing insulin for people that need it. Okay, B. Explain the role of insulin in the body. Very simple. So what does insulin do? So insulin essentially causes our liver to convert glucose to glycogen. Okay, and this occurs when? Well, it occurs when blood glucose concentration is high. And then because it does this, it reduces the blood glucose concentration. Fun fact, you might need to remember this: the opposite, essentially the opposite of insulin, which would be the opposite hormone of insulin, which would be glucagon. And it does the exact opposite, converts glycogen to glucose, right? So glucagon essentially works in the opposite of insulin. It's just a fun fact. And there we go. Some people are unable to produce insulin. This is called diabetes mellitus. People with diabetes mellitus control the condition by using insulin injections, controlling their diet, and monitoring how much they exercise. Explain why insulin is injected rather than orally taken by the mouth. Okay, now the reason for this is pretty simple. Because insulin, as we know, is a protein, right? So when we take in a protein, right, and it passes through our alimentary canal, passes through our gastric system, what's going to happen is it's going to be broken down. Because we have so many protease enzymes. We have pepsin, trypsin, even we have protease enzymes in the small intestine as well, on top of the stomach. So there's a lot of ways that it's going to be broken down, which is why we need to take it via injection. Say why people with diabetes mellitus need to monitor how much they exercise. Well, the reason for this is because exercise actually uses glucose. This increases the glucose use, and this increases the use of glucose and reduces our blood sugar levels. And for people with diabetes, their blood sugar level might become too low because as you can see here, they have to control their condition via insulin injections. They have to inject insulin. So their blood sugar could get very low because they are exercising and using up their blood sugar levels. So exercise lowers blood sugar levels, right? This could lead to a condition known as hypoglycemia, right? Low blood sugar, and that could be problematic. Okay. State how people with diabetes mellitus need to modify their diet compared to people who do not have it. Now, how else can you moderate it? Well, it should be pretty self-explanatory. Even if you don't do biology, you should know that people with diabetes, they have to control their intake of sugars, their intake of glucose, of carbohydrates. So control carb slash glucose slash sugar, however you want to say it, intake. Okay, intake in their diet. Okay, and there we go. That should be it. It is. So yeah, that was a long paper. Towards the end, my handwriting started falling away, but that's why I said everything as well verbally, so hopefully you understood every point that I made. There are a few questions that I skipped over because I didn't feel like my explanation would be much help to you. I felt like it was just a question where you had to work on it yourself, and that's the best way to understand it. And because of that, that's why I want to leave the paper and mark scheme in the description of the video, so you can attempt the questions that I skipped over as well by yourself as kind of a challenge to see if this video truly helped you, see if you understood how to do the paper better as a result of this video. And if you enjoyed this type of content, please let me know. I'll try and make more of it. It is quite a time-consuming video to make, but I will do more if it's helpful. I really hope you enjoyed the video. Please request me what you want to do, what you want to see. If there's any help you need, I will help you out. Just please let me know in the comments. Please subscribe and like the video, and I hope you enjoyed it. See you guys in the next one.